My name is Paige. I'm a PhD student in Dr. Miller's lab, and I'm going to be talking about one of our projects called Developing a Molecular Degrader Against Misfolded TDP43. So TDP43 in a healthy neuron is a protein that's found in the nucleus. So it helps the cell process genetic material and make other proteins, and it really lives in that red center of the cell. In ALS, what happens is this protein mislocalizes, it leaves the nucleus and forms these clumps called aggregates in the rest of the cell body. So this is a double-edged sword because the leaving of TDP43 from the nucleus causes a loss of function. So it's like the manager leaves the office and all hell breaks loose. We lose the function that it's meant to do in the nucleus. But it also causes a gain of function in the rest of the cell, right? These aggregates aren't supposed to be there, so these clumps of protein are disrupting the normal healthy function in the rest of the cell body. So Dr. Miller mentioned earlier that 90% of ALS is singleton, and then 10% is familial. So TDP43 can be caused by, proteinopathy can be caused by mutation in the gene that makes this protein, but this mislocalization is found in more than 95% of ALS patients. So this is a critical question that affects a lot of patients with ALS. So we're focusing on this gain of function mutation, and our goal is to develop a small molecule that can degrade those toxic clumps of TDP43 that form outside the nucleus. So the way that this molecule works is we have one side of the molecule that's an arm that grabs onto the cell's own machinery for breaking down proteins, and then the other side of the molecule is an arm that grabs onto the clumps of TDP43. So the goal is to bring them together so it can kickstart the cell's own functioning of breaking down the protein. So got a little animation to show you kind of how this would work. So the molecule enters the cell and it grabs onto its targets, right? So in yellow, we have our cell's degradation machinery, and red is our TDP43 aggregate. And then it brings them together and lets the cell do its thing and tags the protein and breaks it down. And what's great about these small molecules is that they can be used over and over and over again to break down multiple aggregates of TDP43. So ultimately, we hope that we can use this to reduce the toxic TDP43 aggregation throughout the rest of the cell body. So ultimately, far down the line, the goal here would be to develop this into a drug that can be used to prevent the death of these cells in ALS by reducing the toxic TDP43 aggregation. But I know people have mentioned a lot of different proteins already today, so we would look at seeing if we could swap out that red TDP arm to see if we can grab some other proteins and let the cell get working on breaking those down as well. Short and sweet, thank you.